Thank you very much for the invitation. It's really a pleasure to be here. I've gotten to go to a number of lectures here over the last couple years, so it's fun to get to talk back. I brought a lot of stuff. I didn't bring a lot of stuff from the molecular lab. I really focused, as Pam asked me to, on clinical trials and sort of where we are, where we might be going, and tried to take an approach that mixed realism and hope. Before I launch into that, I think it's important to note that in order to test treatments, at least if we're talking about medications, at some point you end up working with the pharmaceutical industry. So I've had some funding from the pharmaceutical industry. When that is relevant, it'll pop up in red on the slide. And then because I'm giving sort of a broad conceptual talk that I hope sparks discussion, I'm not going to be able to spend the time to call out everyone who's done every individual thing. So these are some of the folks in my lab, but I'm going to pull in stuff from the literature as well, not just things that we've worked on. So as we think about developing treatments, I thought it was important to start briefly with a discussion of autism or autism spectrum disorder, and specifically the challenge of heterogeneity, which is a real conundrum as we move forward toward potential treatments. And then talk about what we know, but really talk about what we know from the perspective of what are the challenges, what are the things that we've learned in order to get information about treatments that work now, or that we know work now. And then I'm going to pivot to talking about how we might get to new treatments. This is more forward-looking, things that we don't yet know will work, but talk about that from two perspectives. The first is really anchored on molecular targets, so this is sort of bottom-up, usually gene, all the way up to potential treatment, sort of rational therapeutics as we think about it now. And then the other angle, which in some ways is top-down, symptomatic targets, sometimes with knowledge in hand about how those symptoms might be mediated at a circuit level, sometimes without a lot of knowledge in hand. And then talk about how we might go from where we are now, which is these narrow groups, 1% or less, that share a common risk factor that contributes the majority of their risk, to perhaps larger groups unified by some other biomarker approach. And finally, to come back around and recognize that we're not going to have magic pills that, at least not in my imagination, that by themselves cure autism, but that what we're likely to have is medications that may put the brain in a state better able to benefit from behavioral or educational interventions. So I think it makes sense to start with humility. So what is autism spectrum disorder? I guess the short answer is we don't really know what autism spectrum disorder is. We're describing it on the basis of behavioral symptoms. As Isabel Rappin said in a class that I got to sit in on a couple years ago to a group of postdocs, autism is not a disease. It certainly is not one single thing. It's a description of a common set of outcomes in people who probably have many, many different paths to get to those outcomes. And those outcomes are described here. I'm not going to spend a lot of time talking about what the core features of autism are, but suffice it to say that I really never have seen somebody who only has autism spectrum disorder symptoms and really doesn't have any other difficulties, doesn't have anything else that's relevant as we're having a conversation about how to approach treatment. There are groups of people who fit different patterns. This pattern fits actually a lot of the folks with rare genetic syndromes or rare copy number variants, now increasingly single nucleotide variants, that we understand as leading to a lot of autism risk. And so oftentimes you also see intellectual disability or at least some cognitive decrement, language impairment, sometimes seizure disorder, oftentimes abnormal EEG, sometimes other indications that there's a syndrome going on. And then this is just one example of another group that may share a common biomarker, a different pattern of medical difficulties. And when we do a study of autism treatment, or frankly, a study that's defined by quote unquote autism spectrum disorder, we're taking people who fit this pattern, and 100 other patterns or 150 other patterns if it's a well-powered treatment study, and lumping them all together to assess if a treatment works. And we need to be honest about that approach. That doesn't mean it's stupid. It just means we have to approach it understanding that that's what we're doing. When we think about autism risk, a lot of the epidemiology still fits this model where you have multiple common risk variants, either genetic or environmental. We know, actually interestingly, more maybe at this point about common environmental risk than we know about common genetic risk. But that when those variants occur together in an individual, they'll lead to increasing risk, probably in some sort of interactive fashion. But what's actually emerged in the research, primarily on the genetic side, and a lot of that sparked by the Simons Foundation, are quite a number of rare things that individually contribute the large part of risk for an individual. Most of these are not deterministic. So Fragile X syndrome is the one of these that's sort of been around the most and connected for the longest time. And in Fragile X syndrome, it isn't the majority of people that have autism, but you do find Fragile X syndrome in about 1% of people with ASD. Chromosome 16p11 deletion or duplication, we now understand to be a major risk factor for autism, but again, it's the minority of folks who have autism spectrum disorder. And then we have things like tuberous sclerosis, P10 hamartoma syndrome, first thought of in the medical or neurological fields, and then later understood to increase autism risk, and increasingly things that are identified within large populations of people with autism, not on the basis of initial suspicion medically or as part of a syndrome. Some of these are at this point logically targetable. So a recent example we were talking about earlier from last week in Cell, a paper that suggested a treatment that could be administered in a mouse and rescued difficulties in a mouse, but a very logical translation straight to the human. Not necessarily the sort of thing that would lead to extensive clinical trials, though, because you have 2 or 3 individuals in the world who've been identified. And then we have some environmental variants, environmental risk factors that have a similar pattern. So fetal valproate exposure is one of these. What we would hope over time is that these cluster, and there is some evidence of clustering. Ideally, if they cluster, you would have not just a cluster of rare variants that affect the same pathway, but hopefully that pathway would also be implicated in some people with what's now termed idiopathic autism, meaning autism where we don't know that there's a rare variant that contributes to the diagnosis. The genetics, unfortunately, is much simpler than what comes next, at least in terms of numbers. So part of that complexity is development. We always have to maintain a developmental perspective. But as you move upward from genes to proteins, protein networks, as you move towards cells and synapses, you move from tens of thousands of genes to quadrillions of synapses. But then you should see convergence further up, so that if we're selecting our population on the basis of behavior, you would imagine there might be some divergence at the genetic level that then spreads out further when we're thinking about some systems at the level of cells or synapses. We should see convergence under the behavior again. You should have some systems in common. We don't yet have a ton of evidence for that, but what we would hope for is that some of these rare things that individually maybe contribute to autism in 1% or less will point us towards systems that are involved in the larger group. Most of the actual convergence at the level of data thus far has been lower down at the level of protein networks that may be targeted, but with a lot of uncertainty about how that rescue at the level of protein network might affect behavior in a human. And that leaves us with a considerable black box. That black box is particularly challenging at the level of studying outcomes in humans. I would say at the same time that's a lot better than where we were a decade ago, where there wasn't a lot that you could say that was rational about potential autism therapeutics. So you could think about things that work for other disorders and try to adapt them, but without a knowledge of neurobiology. So I would say really that's quite hopeful, but there is an additional challenge. And that additional challenge is that over and over again we see that increased dosage of a genomic region or an autism risk gene may lead to autism risk. And then you find shortly thereafter that decreased dosage also leads to risk, either of autism or another developmental disorder. In a certain sense, maybe that makes sense. If you're thinking about wiring a system, it doesn't matter if you miss this direction or that direction. You really need to line things up perfectly. That's sort of cartoonish. I think at this point, we don't know whether we'll be able to target the same circuit, say, in both groups, or whether it's more like if you move this person closer to health, you move this person actually in a worse off state. So moving from that, which is, I realize, a broad cartoon of autism risk and how that might be realized, I wanted to talk briefly about what we currently know about autism treatment. So this is about what works, but also about how we know that it works. On the behavioral side, and this is where we think most of our efforts should go, we know with some certainty that early intensive behavioral intervention makes an impact. What it impacts on specifically seems to be cognitive outcomes to a larger degree than language outcomes, and then some impact also on adaptive behavior. You'll note that none of this says autism symptoms, and that's because that's been much harder to see in these studies. There are other targeted behavioral interventions that are really for specific symptom domains, so I don't want to say that's the only area of research evidence. But this is one example from the early intensive behavioral intervention literature. This is actually the Early Start Denver model, which is a modification of this sort of behavioral intervention that uses more play. But what you see here is that there is a significant improvement in IQ scores measured over the course of treatment. And you'll note that that is significantly better than what's seen in the comparison group that is receiving treatment in the community. You note that actually this is only at a trend level, but there is a trend improvement in adaptive behavior. But what that represents actually is a failure to lose against peers, which is an improvement, an ability to maintain position within peer group, which is great. But it's important to note that this sort of outcome measure isn't what we typically use in a study of a medicine, right? You would never study a medicine, test IQ before and after and say, okay, that's our outcome measure. So we have to be very careful in taking the right lesson from this literature that tracking cognition, which is oftentimes tracking along with autism, may be one way to have effective outcome measures. So on the medication front, we know that some medicines help, but no medicines are known to help with core symptoms. And so the medicines with the best evidence are risperidone and aripiprazole in a class that was originally described as major tranquilizers. These medicines really should come into play only in the minority of children with ASD. They help with what's described as irritability and agitation. Really, we're talking about aggression, distress, tantrums, self-injurious behavior, things that really merit using medicines that have a significant side effect profile as well. When you look at those medicines over time, in the initial studies, you saw actually very little response to placebo. But in the follow-up studies, as people started to believe that medicines could help with these behaviors, you see a significant improvement with placebo. So this is one of the more recent studies looking at aripiprazole, or Abilify is the brand name, and you see that the placebo effect improves pretty substantially. So that the active medicine, and there are 3 active medicine groups here at different doses, improve not even twice as much as the placebo improves. And you can imagine the challenge with that. This is a substantially sized study, so this has more than 200. So this is the placebo group right here at the top. And then these 3 are the active medicine groups. So you see that they are statistically better, but even with something that is as active as a major tranquilizer, it becomes hard to separate from placebo once you have this level of effect. And large sample size. So that behavioral treatment study, the sample size in each of those groups is 24 individuals, and here we're looking at 50 individuals per group. So much larger sample, easier to detect significant effects. I'm very briefly just going to note that there's good evidence that medicines that help with attention deficit hyperactivity disorder also help with those symptoms in children with ASD, so I don't want to neglect that. It's not terribly interesting to spend time pulling that apart. But this is another example from what's already been done that's instructive about how we carry forward. So repetitive behavior is part of autism criteria, but we don't have evidence that the repetitive behaviors we use to diagnose autism improve with any medicines in particular. There's a history there we could talk about afterward, but there is evidence that serotonin reuptake inhibitors like fluoxetine help with obsessive compulsive symptoms in people with autism in adulthood. Okay? There was a study looking at the same medicine, fluoxetine, in children that failed, was never published. And then there was this study of citalopram that lumped all repetitive behaviors together and looked to see if kids improved. The initial report was, it's no better than placebo. But then they followed it up, and this is work from Brian King and a larger group of colleagues. They followed it up to understand maybe what was happening with their high placebo response, and they did see a substantial response to placebo. And they did median splits in a number of symptom domains. So one of those symptom domains was disruptive behavior. And when they did a median split, so they looked at the group that had less disruptive behavior, the group that had more disruptive behavior, they found that there was a significantly higher response to placebo in the group with less disruptive behavior. The same was true when they lumped together mood and autism withdrawal sorts of symptoms. Again, bigger response in those who are less affected. And again, doing the same thing, just looking at caregiver strain. So Allison's giving this look like, what the heck? But this is one of the challenges when you move into a clinical trial, lumping all repetitive behavior together. Some of these families were not very distressed by their children's repetitive behavior. The repetitive behavior likely wasn't interfering a lot with their day-to-day life. Those families were more likely to see improvement. The clinician was more likely to say that that child was much or very much improved. The families who were more distressed, the children who are more distressed, were much likely to see benefit with placebo. And if you look at it, maybe there's even a signal under here, but it's a little hard to tell, meaning that maybe there is an advantage to active treatment over placebo in the kids who come in with more distress. But that's not the way the study was done.
What is lower and upper?
So this is it's a median split. So these are kids, the lower half of symptoms of disruptive behavior or the upper half of symptoms of repetitive behavior. Yeah. More disruptive behavior, and more mood symptoms, more caregiver strain. So that may be also useful for us when we're thinking about moving forward. And then the other example is actually, in a lot of ways, a discouraging example. So the medicine that's actually been studied most in autism spectrum disorder with most randomized controlled trials is a medicine that leads to no benefit whatsoever, which is secretin. And this was studied based on a child who had reported improvements after a GI procedure where they had an infusion of secretin. About 20 years ago, there were probably thousands, tens of thousands of kids in this country that received secretin infusions. It leads to no benefit beyond placebo. But what emerged from these studies is that the placebo led to significant benefit across a wide variety of ratings, whether by the clinician or by the parent. More so by the parent than by the clinician, but the clinicians saw significant improvements as well, such that in some of those studies, the placebo improvement was 50%. So beating 50% improvement is pretty hard when we have very narrow slices within autism where we think we understand the pathophysiology. So this is an example of the most prominent of those studies. A reasonable sample size, not huge, 30 individuals. This is the response to placebo right here that you see over time. And this was on a number of measures. I just picked one that was a composite measure of autism symptoms. And this is the response to secretin. So placebo outperformed secretin, but placebo did really well.
How do you explain that?
So that's where we're going next. So some of these are parent report measures. None of them really were the child's report of their experience, but some of them also were clinicians rating the child's improvement based on interacting with the child and based on the parent's description of how things were going at home. Yeah?
So do you think this is placebo effect on children or on caregivers?
Let's get there. And this is one of the things that came out as part of this. After this study was published, people sort of latched onto this one quote that was included in the study, which is that 69% of the parents, after they were told about the results, said that they were still interested in secretin as a potential treatment for their child. And that doesn't mean that parents are somehow deluded. It means that we all hold on to hope that if it was done a little differently, it might work better. And we have to be really careful about that bias. It's an inherent bias toward hope and toward the desire for a treatment that makes an impact. This is a slide of the overall impact of placebo in trials across autism, also from Brian King and colleagues. And you can see that it hovers around 25 to 30%. If you spend more time with it, you find that those studies where there was more side effect associated with the drug had a lower placebo effect, probably because the clinicians figure out who's on drug and who's not. And those where there were fewer side effects associated with drug have a higher placebo effect. So why does this happen? So there are a lot of reasons. So the one that we know the most about, actually lots of research done over time, is the reality of a placebo effect. You can actually tell somebody you're giving them a placebo, and you will still see a placebo effect. Isn't that bizarre? But it's true. So we know a lot about this over time. So it can move things you'd expect it to move, like pain, but it also will move bold signal on an fMRI. It can move blood pressure, cholesterol. If you are doing an endoscopy to look at esophageal ulcers, you will see that placebo can improve that as well. Response increases with a number of things. So intensity of treatment. So if I'm doing a sham surgery, I'll see a larger placebo effect than if I'm giving you a pill. Cost of treatment, more expensive things, bigger placebo effect. Probability of active drug, and so that probably went into that aripiprazole study I showed you. They knew that there was a 3/4 chance that they would get active drug. You're much better off having a 50-50 chance. That's much more where people will back up and say, I could be on either. Subjective outcomes, so that makes sense. So if you just ask somebody how they feel, that's something that's going to see a larger placebo effect, say, than ulcers. And then inverse illness severity. So the more severely ill somebody is, the less robust the placebo effect is. In behaviorally diagnosed disorders, things like obsessive compulsive disorder, trichotillomania, hair pulling, have less of a placebo effect than things like mild depression. So when we describe placebo effect, it's not all just that, however, right? So really we're talking about non-specific changes that occur during the trial period. So it isn't all just the effect of hope, although that's certainly some of it. Some of this is really straightforward, right? So the reason all of us think that criticizing our children or nagging our children works is because we pick this point in their behavior. We all fluctuate, right? So if you pick them behaving at their worst and bug them about it, chances are when you next look at them, they'll be behaving better, and that reinforces our behavior. But that also can happen in a clinical trial. You don't enroll when things are going well. You enroll when things are going poorly. Continued development is also part of it. So we're studying a neurodevelopmental disorder. Development doesn't stop because you have a neurodevelopmental disorder. It may follow a different course. And so if you're asking questions about neurodevelopment, is this child learning more words? Unless you have a clear comparison for how they were before or how they would be without the drug, it's really hard to tell. So shifting from here to here might be the goal, but it's actually quite hard to detect, at least subjectively. And then sort of like the uncertainty principle, if you are asking all of these questions over and over again, it can actually change things. So this is an example from the Vineland, which is actually an anchored scale. It's better than many of our scales. If I say to a parent, can your child point to at least 5 minor body parts? The first time I ask it, they may say, without the child in the room, I'm not real sure, maybe some of the time. But you better believe they're going to go home and check. And the next time they come in, they'll tell me, yeah, they can. But that isn't because anything changed, it's just because we got them to check. And there are lots of examples of things like that. You also oftentimes ask parents to compare against peer group. And so that can shift over time, or a parent can leave and then come back and think, I've paid more attention to peers, ratings actually change. And then there's the effects of care. And this isn't placebo. This is the effects of being in relationship with a caring provider, and you would hope that the clinicians in these studies are caring providers. This can be coaching, subtle or not. Have you tried this? Have you tried that? It can be modeling. So in the course of testing, you may use a technique that the parent sees and then jumps on top of, and that can lead to improvement. And then encouragement. So she really seems to be talking more. The parent then is going to try to engage more, might be reinforced by that, might see more conversation, not because of the effect of a drug, but because of the encouragement that we've given. So how do we mitigate this? So one thing is, as much as possible, always have a control. And if possible, always an active control. So if you're doing a behavioral study, waitlist control is the opposite of placebo. Right? It's saying you're not going to change until you get into the active treatment in 3 months. And so we need to be really hesitant to interpret those studies that have waitlist control initially. Sometimes it's necessary, but then it needs to have the active control moving after that. You want truly blinded raters. So if your medicine leads to a side effect, so with risperidone, sedation is really common at first. That's a tell. The clinicians are going to know what they're taking. Bumetanide, so there have been some really exciting recent papers on bumetanide, but it leads to a lowering of potassium levels. So if the clinician is seeing that, they'll know who's taking medicine. So you then have to have a blinded rater. And you want to make sure that the parents also don't see the results or don't know about the results. Longer duration of treatment. So if regression to the mean is part of the story, if you follow over a longer period of time, you get a clearer sense of whether it's a real change or something that's part of fluctuation, and then equal likelihood of each treatment. You also want to have outcome measures that are as objective or anchored as possible. So IQ is a great one, right? There's not a lot you can move there, except that your expectancy bias can still have an effect, and if the child has been trained to sit still and work, they're going to do better. So you have to even be careful there. Things like the Vineland that are anchored are much better than things that are totally subjective. And then we have things that are really ill-defined but capture what we do as clinicians, like clinical global impressions, which can be useful for saying if this is a meaningful improvement, but don't necessarily tell us what moved. Finally, as you're choosing your outcome measure, and this has been a real challenge in things like Fragile X syndrome, you have to find a spot in the outcome measures distribution where you actually can see movement. So if somebody is bottoming out at baseline, it's very difficult to see that you're going to have significant improvement that separates from placebo, whereas if they're closer to the middle of the measure, whatever the measure is, you're more likely to see movement. And in some ways, the same thing is true at the top of a measure. So if people are very severely affected, so that everyone is at the top, it's hard to be confident that you'll move. So one approach that I like better is to actually not tell the clinician or the study participant when you're switching, but to switch more than once. So to not just put somebody on active medicine and at the end take them off versus somebody who's just put on placebo. Either you can do a blinded, meaning the clinician doesn't know, placebo run-in, so that you are able to observe over time before you end up giving active medicine, or you do on-off-on so that you establish stability of behavior. And this is an example where this individual started on an intervention, and this is actually from the behavioral literature, because this is how behaviorists know that something works for an individual child. So intervention, you stop the intervention, you see that they establish a new baseline, start the intervention, see a new baseline again, stop again. This is how you know in the clinic that something's working. This is how we test medicines all the time as clinicians. So this is when I'm most confident a medicine actually works for a child. When we stop it and things get worse, and we start it again and things get better again. It's not perfect, it's not as good as a controlled trial, but if you do that across multiple individuals and you see the pattern emerge, you can control better than we do right now. So that's sort of the bad news before what I would say is the good news. And the good news is not as good as it might be, but we're getting there. So I would say that this precision medicine approach where we have rare genetic risk factors for a disorder leading to molecular targets is going to get us toward meaningful treatments. I could talk about that in a number of contexts. I'm picking Fragile X syndrome today because it's one where we've made a lot of progress, but we're not really there for any of these yet. This is really still forward-looking. So Fragile X syndrome is something that leads to autism a minority of the time. Depending on your lines, maybe it's around 50-50, depending on your lines, depending on the study you like, always leads to mild or moderate intellectual disability. Also leads to symptoms like ADHD, can lead to sensory sensitivity, higher risk of seizures. These kids are in many autism populations, usually are pulled out because they look kind of like me, with large ears, prominent jaw, and prominent forehead. So we've now known about the molecular genetics of Fragile X syndrome for 20 plus years, 25 years. And I'm going to move through it very quickly because I want to use it as an example, but I don't want to neglect it completely. So there's an expanding trinucleotide repeat in the 5' untranslated region of the FMR1 gene. That leads to gene hypermethylation, essentially shutting off the gene. for further transcription. It encodes FMRP, which is an RNA chaperone. I'm not going to talk about that role. I'm just going to show a cartoon of one understanding of what it does at synapses. So this is a glutamatergic synapse, where you usually think about glutamate as hitting AMPA receptors or NMDA receptors, but it also acts on metabotropic glutamate receptors, and that's where FMRP seems to lie in this cascade, where downstream of mGlu5 signaling, you see translation of more FMRP, but you also see that it acts to dampen down further signaling, and that further signaling serves to internalize AMPA receptors or remove AMPA receptors out of the recirculating pool, so that if you don't have FMRP, you're lacking the release on that brake mechanism. You have too little AMPA receptor at the cell surface. You get excessive long-term depression and failure to form mature synapses. When that happens, you can actually see it anatomically. You, me, I don't know, but people can see it anatomically. A lot of this work was mapped out by Mark Baer's lab, where they were able to show, Steve Warren's lab did some of this, and a group at UIC, I'm not going to remember his name now, did a lot of this, showing that you can see excessive nearby dendritic spines because you're not suppressing them with a mature synapse. You can see that here. It looks nearly identical in the mouse model and in the human pathology. So this is beautiful face validity, says that this really does differ across species. Unfortunately, if you observe the mouse, and people have done this over years, it has some mild hyperactivity. It has some fairly subtle learning deficits that don't translate easily to the human. And its social behavior is not much different than a typical mouse. It backs out in the tube test, but otherwise it's pretty equivalent. So it isn't something that maps easily onto symptoms. It takes us to something that looks maybe like this, all the way up to synapses, and then a gap before behavior. Maybe our black box actually even includes behavior, because we don't know that targeting this system at a synaptic level will impact a particular behavior we study in humans. So the exciting thing is, at least in a mouse, compounds that can be studied in humans rescue every observable phenotype. So this is work that was done at Roche in collaboration with Mark Baer's group, where they were able to show a rescue of neuroanatomy, a rescue of behavior, a rescue of learning by treating mice essentially shortly after weaning all the way through sexual maturity. So that's 4 weeks in a mouse. Obviously a lot more complicated when you move to a human, but essentially rescuing everything that we can observe that's different in a mouse. So now you need to carry that forward, obviously, into a human. And unfortunately, because mostly regulatory expectations, you need to start in adults and then move to adolescents and then move downward. And both Roche and Novartis dropped their programs after results that look like this. And these are the published results from Novartis. Roche's results aren't published, but in gray here, and I realize that the gray is a little hard to see, so I'm tracing it, you can see that in each graph, placebo is either better than active treatment or at least not worse than active treatment. And that was true in a number of different ways of slicing the data, seemed to be true in adults, seemed to be true in adolescents. So that feels discouraging, except that this is never the way that you would have designed this study 20 years ago, right? This is what you do because regulations say you have to start in adults, and because in animal models, surprisingly, you can rescue a lot of things in adults. But what you'd really like to do is to move into kids. Now these programs have closed, but we need to answer some questions before giving up. So there is the question about placebo effect and whether that dwarfed the impact of the medicine, but placebo shouldn't outperform medicine. There's a concern about target drugs, but I would say ultimately the concern's probably more about outcome measures, whether we're really looking at something that we would be able to observe different in a mouse, about the ages of the individuals studied. So this is a neurodevelopmental disorder. Humans need to learn a lot more between weaning and sexual maturity than a mouse does. So studying what moves in a mouse in adulthood is quite different than learning what happens in a human. So there's now funding from NINDS for a child study led by Liz Berry-Kravis of the Novartis compound. And that child study is seeking to carry forward, basically to shine a light in the middle of this black box, both in highlighting development, but also looking at cognition as one outcome and looking at circuits using EEG and ERP to try to understand whether you're actually moving something regardless of whether or not you're moving the sort of behaviors we use to diagnose autism. There are other targets we could talk about. I'm not going to spend time on that, but I want to note that at least to this point, it doesn't look that different than the initial cascades in cancer. So if you look at the HER2 new receptor and the development of Herceptin, it took a decade and a half to 2 decades to get from an observation to something that was tested in the clinic. Now in cancer, they have much easier outcomes. And that's not different really than the time course 1991 to when we started to initially have trials in Fragile X syndrome. I can't tell you exactly when this is going to pan out, but I think that there will be a treatment that will have an effect based on the neurobiology. What does that really give us? So first thing is it gives us a treatment that would work in Fragile X syndrome or another rare genetic risk factor for autism. It importantly also gives us a way to understand how to approach treatment across neurodevelopmental disorders. So if we found an outcome that works in Fragile X syndrome, maybe that also works for folks with CHD8 mutations, right? We need to know how to study this as much as we need individual areas where we can see improvement, and then that should crack things open. And then there's also the possibility that this will extend to a larger group of individuals who don't have Fragile X but have similar pathways implicated. So I'm briefly going to talk about symptomatic treatments. So this comes out of the realization that we don't really have any rationally based therapeutics that target disorders that are diagnosed based on behavior. That's a long way of saying in psychiatry, we honestly really don't know why most of our drugs work. In neurology, we have a little better idea, but usually for things that target, say, epilepsy. And most of our treatment success is not around autism specifically, it's around things that are really concrete. So when a kid comes in, I know I can improve their constipation. I know treating their epilepsy, referring them to a neurologist is the first priority. I know that there are interventions that target language. I know that there are behavioral interventions that lead to improvements in IQ. None of this is autism, right? But it all matters quite a lot. I know that we have medicines and behavioral interventions that improve associated behavioral symptoms. And then we've got these things like sociability, where that's not in the definition of autism, right? So somebody with autism can really like being around other people, but can struggle in interactions. Autism is really defined by criteria that are much more around social competence and social communication than about preference for other people. So I could make somebody worse with something that made them more sociable, but I might make somebody else a lot better. These sorts of treatments, I would say, could impact a larger percentage of folks with autism spectrum disorder, but they're not likely to be home runs, right? So this is much more like a base hit, but base hits are good. We don't have a lot of base hits. And so I would say that this is absolutely an approach that's worth pursuing. The hope is that you pursue this approach by understanding something about the circuitry that underlies particular behaviors. The truth is, as I told you, almost all of the time we're occupying more of a black box world. Oxytocin is one example where we do understand something underlying social behavior across species. Again, I'm using this as an example, so I'm not going to talk about the neurobiology in detail, but suffice it to say that across species, from rodents to humans, oxytocin is implicated in social function, particularly sexual function and maternal function, but also things like trust can be moved by brief exposure to intranasal oxytocin in a laboratory setting. There are also lots of caveats that we could discuss afterward. In autism, however, you would not say that we have clear evidence that oxytocin is implicated in the pathophysiology. There are a few emerging areas where maybe there's clearer evidence, but overall the data are quite mixed. But in a laboratory setting, you can change behavior in somebody with intranasal oxytocin, whether they have autism spectrum disorder or not. But this is an example in autism spectrum disorder where people with high-functioning autism spectrum disorder were compared to typically developing controls. And you can see that the ASD group either got a placebo nasal spray or an oxytocin nasal spray. In this cyberball game, you're throwing an imaginary ball to what you think is a real person who either always throws it back, throws it willy-nilly, or never returns it. Most typically developing individuals prefer to play catch with the person who throws the ball back. Most folks with ASD didn't have that preference unless they received intranasal oxytocin. This is not autism treatment, to be clear. This is change in a particular behavior in a laboratory setting. But this is a study that followed soon after that French study where Kevin Pelfrey's group at Yale looked at potential change on fMRI after receiving intranasal oxytocin. Interestingly, they did not see a behavioral change, even though they hoped to. But they did see a change in fMRI bold signal such that if you received intranasal oxytocin, you had enhanced signaling in a number of regions when presented with social stimuli with eyes, and actually less when presented with vehicles. That included areas like the nucleus accumbens, which is important for reward and has been implicated specifically in social reward, as well as a number of regions that they describe as part of the social brain, which I think is reasonable, except that it really is pretty broad activation. So this is not something that is ready to translate in this way, but I updated this just yesterday. It continues to be a different brand every few months, but intranasal oxytocin or oxytocin nasal spray has been available online for at least the past several years. Who knows whether it actually contains oxytocin, but there are probably thousands to tens of thousands of children with ASD in the US right now receiving something that is at least marketed as intranasal oxytocin, and the real thing can be gotten. There is still obviously a big gap here, but as I mentioned, there is some emerging data pointing to the oxytocin system in mouse models related to ASD, including one model that goes all the way up to showing a developmental impact of oxytocin in early life, leading to improved function of the oxytocin system later and improved social function. So there is an emerging neurobiology coming out of autism mouse models as well. Really, though, it needs to be brought into the real world. We need to see this applied with outcomes that really matter. We need to see if this makes a difference when given more than once. I think over time, we probably need to put it together with behavioral interventions. That takes us to, I realize I'm going to be running a little bit short on time, but that takes us to the quandary of wanting to move past the 1% and also wanting to move past things that are purely based on symptomatic treatment or that black box. And that would fit sort of in between, ideally with a biomarker that would identify a larger group that might share common pathophysiology. And so I'm going to use one example of this. I don't want to say that this is a perfect example, but it's an example I use because it's what you would expect if you had a treatment that benefited a subset of kids, but you were studying all of autism. And that's a real challenge because that's exactly what we expect. So this is an example of our baclofen. If I use this figure, this is mostly based on excitatory-inhibitory imbalance as one potential risk factor for autism, and that's really a swath at the synapse. There is other evidence we could spend time talking about. And these are the results of one parallel group randomized controlled trial. One group is taking placebo, one group is taking baclofen. The primary outcome measure here was social withdrawal, which you see didn't move. It's not a great outcome measure. It was picked because it fits what the FDA is looking for. Paul could fill us in on how that process went. But you see that it did move one of the outcome measures right at the threshold of statistical significance once corrected for multiple testing. And that's a clinical global impression of severity score. So just to show you that it really didn't move the primary measure, this is change from 0, from baseline in the primary measure. You see already at 4 weeks, you see a substantial placebo response that's nicely matched in the active treated group and no difference also at 12 weeks. So not what we were certainly hoping to see. But then you see this clinical global impression of severity also shown as change from baseline. And you see already at 4 weeks, there is a change in the placebo group. But by the time you hit 12 weeks, there's a significantly greater change in the group receiving our baclofen. Really, this is the smallest sort of outcome measure you can have. It's really 7 points. But you start off with 0 change, and you can see there were more people in the R-baclofen group, more people in the placebo group that had no change from baseline. And as you move over into these larger changes from baseline, you see that there is a substantial increase in the R-baclofen group. But these are big changes, and these are small numbers. Okay, so this is sort of a basic small numbers problem where it's hard to pick out exactly what that subgroup is. We, and Paul was one of the lead authors on this paper, tried to pull out individual subgroups. Paul did much of this when he was still at Seaside, but you see that there was a bigger advantage for arbaclofen over placebo in children than in adolescents. All this is post hoc, so I want to make sure we see it with a grain of salt. Bigger but quite variable response in kids who are more mildly affected. This is before DSM-5, so autism spectrum disorder versus autism. And then if you look at individuals split by IQ, you see similarly those with an IQ over 70 tended to have a bigger response to arbaclofen versus placebo. So that may be some clue about how to carry forward. There also, for the Vineland Adaptive Behavior Scale, was an intriguing post hoc analysis that Paul led, looking at individuals who had the same raters across the study. This, of course, also drops out those kids who were unable to complete the study for one reason or another. But there, if they had the same raters for the socialization subscale, you saw a significant improvement on arbaclofen versus placebo. This is something that is much better anchored than most of the other measures in studies like this. So things like uses action to show happiness or concern for others, always, sometimes occasionally, or never, gives you a much clearer sense, a much better anchor than a lot of our subjective, how sociable is the child. So from this initial study, you have to be hesitant, right? This needs to be replicated. I think really carrying it forward, you'd say it's not going to be most likely a general treatment for every kid with ASD, but I think that that's true of almost every treatment that we might identify. The clinical profile, I would say, is useful, but ideally you'd like a biomarker that targets what we think is potentially going on, something like this where you are able to probe a particular circuit, see your drug is engaging that circuit in a child, perhaps in the context of a sensory or cognitive task, and then look to see if that same child sees improvement in behavior over time. There are lots of biomarkers that we could think about. I think of biomarkers in ASD in a number of different ways. One way, and I think maybe the most important way, is to think about subgroups to target for treatment. Because it is so heterogeneous, I think we need to find ways to unify subgroups. So one example of that would be on the genetic front. So there are a number of significant enhancement of genes identified in ASD that are bound by the Fragile X protein. So, yeah, upstream actually. So FMRP binds the mRNA of those corresponding genes. Now there's question because you don't necessarily know whether the effect of FMRP binding is to move it up or down, and oftentimes the mutation that's identified in autism is disruptive, so there's still that direction question. But if you could identify directionality and identify that this is a gene that's disrupted that's bound by FMRP, maybe that individual would benefit from something that helps in Fragile X syndrome. I would say in neuroimaging, for me at least, because I work with kids who are more significantly affected typically, it's hard to think about putting kids in an fMRI paradigm in order to test whether they're going to improve on a medicine, whereas EEG is quite accessible, not for every child, but for more. There are also subgroups defined by overgrowth, either brain size and head size, or overall body overgrowth that appear to be a robust subset. Kids who have elevated cytokines, so one example of this would be IL-6, where there does seem to be a subgroup of kids with elevations. Well, it turns out there is a drug that is an IL-6 antibody. So if there is a subgroup where IL-6 is involved in pathophysiology, that's a logical target right there. There's emerging examples for IL-1 beta and for a number of these others. So that's another place you can think about a biomarker that even connects more readily to treatment. And then I would say most of our environmental risk factors and some emerging genetic risk factors are really acting on the maternal side or acting at the level of the placenta. Seems like mid-gestation seems to be one of those risk periods. But there's emerging findings there like maternal antibodies that may identify a subset of kids that have common pathophysiology. This is still emerging, but I think it's one area of potential interest. And I realize I'm going to skip. This is painful for me, but I'm going to skip past my restricted interest. If somebody wants to ask me about serotonin, I'm happy to talk about it for a while, but I'm going to skip past a few slides so that we get to have a discussion. So if we can identify therapeutics that fit one of these patterns or more of these patterns, I think we have to be cognizant of the need to couple something that affects the brain with something that actually delivers learning. In ASD, there aren't really good examples of this. This is one example that did show a significant effect, but I'm hesitant to try to convince you that it's clinically meaningful. So both groups that you see here rated for severity of disruptive behavior on this side, weeks in the study on this side, both groups are receiving risperidone, both in blue and in pink. The blue group is also getting a parent-delivered behavioral intervention. And you can see that there is a statistical improvement at the end of the study, but it's quite small. There's a follow-up study from the same group, the RUP group, showing that if you deliver the behavioral intervention on its own, it does show significant benefit, but does not show as much separation from the active comparison group as something like risperidone. So this is a challenge when we try to put things together. Here you might say we've hit a floor effect, that there's so much benefit from risperidone that it's hard to show additional benefit from the behavioral intervention. But we need to think about ways to put those two things together. The sort of thing we'd like to see instead is more like the D-cycloserine story in anxiety disorders. So there's a long literature here emerging from rodents where you can show that D-cycloserine potentiates extinction learning, moving into humans initially with very simple anxiety disorders like phobias, and then broadening out from there. This is one example from obsessive compulsive disorder. Both groups here are receiving a sub-threshold behavioral treatment. And you can see both groups do show a significant improvement. The rate of improvement is more significant in the group receiving D-cycloserine. It's a partial NMDA agonist. It ought to improve learning, and it does seem to when using extinction paradigms in the clinic. Not that this is necessarily going to be the answer in autism spectrum disorder, but imagine if we had a treatment that doubled the rate of response to early intensive behavioral intervention, right? That would be the sort of thing that we'd like to see. At this point, I can't say we've got something like that. But that's the sort of thing I think we can hope for if we put an understanding of neurobiology together with an understanding of behavioral principles. So this is my summary slide. So I don't have to tell you again, ASD is not a disease. It's a description of a group of things that we can observe in the clinic. But increasingly, we're able to pull that group apart. We do not have neurobiology-based treatments right now. We have a real challenge getting past the placebo effect. That requires a significant investment, both in large studies, but also in methodologies to get around it. But I think that we do have substantial promise. A lot of that promise is emerging from what seems like the weekly description of a new autism risk gene or risk factor. And I think that that will lead us to targeted neurobiology-based treatments for subgroups of kids that we then can couple with what we currently know about behavioral intervention. So thank you to any number of people. I actually skipped past a lot of the lab stuff about serotonin, but a number of people involved in the lab side, and then also thank you to funders, those directly involved at some level in some of what I've described. And I actually hit 6 o'clock right on, so we have time for discussion. Thank you.